We often picture the past through, fossilized bones or maybe fragments of stone tools. But what if the real story, the details of ancient life, were hiding in plain sight, locked away in microscopic traces of DNA? Welcome to the Deep Dive. Today we're going on a pretty remarkable journey, not really across space, but deep into time. 2 million years back, actually. Our source is a truly groundbreaking paper, a 2 million-year-old ecosystem in Greenland, uncovered by environmental DNA. It's led by Kjær and Winther Pedersen, and honestly, it's changed a lot about how we view the ancient high Arctic. So our mission here is to unpack the methods they used and, crucially, the discoveries they made. We want to figure out how they got genetic information that old, what kind of lost world this DNA points to, and why this ancient ecosystem is actually really relevant for the climate challenges you face today.
Right, and to really get it, we need to understand the time frame. We're talking about the late Pliocene and early Pleistocene periods. So that's roughly, say, 3.6 down to about 0.8 million years ago. And geologically, this is super important. Why? Well, because the climate conditions back then, they look a lot like what models are predicting for our future under, well, significant global warming. It's like a natural experiment already run for us.
A past analog for the future.
Exactly. And during that time, we know polar amplification was really strong. Evidence points to Arctic temperatures being, get this, maybe 11 to 19 degrees Celsius warmer on average than today.
Wow, 11 to 19 degrees warmer.
Yeah. So if we want to understand how life up north might respond to future warming, we really need data from when the Earth was already that warm or even warmer.
And the specific location for this study, it's the Cap-Cuban-Have Formation. That's in Land, way up in North Greenland.
Way up there.
If you look at it on a map today, it's basically a polar desert. It sits at 82 degrees north latitude. It's incredibly harsh, very barren.
But it wasn't always like that.
No, exactly. Earlier geological work there, over the last maybe 40 years, it hinted at something much richer back then. It looked like it might have been near the boreal Arctic ecotone.
Uh-huh. That transition zone.
Right, the boundary between denser forests and the open tundra. But those early reconstructions, they were based on pretty sparse fossils, some pollen. It was a fuzzy picture. Suggested maybe minimum winter temps around minus 17 Celsius.
Yeah.
But the full complexity, totally hidden.
And that context matters because those warmer conditions, they must have involved huge melt events. Significant melting of the Greenland ice sheet.
Which has implications today, of course.
Absolutely. This period was likely one of the last big ice-free times for Greenland in the last, say, 2.5 million years. But, despite finding the odd macrofossil, maybe an insect bit here or there, we just didn't have the resolution to see the whole community.
Okay, so that leads us to the big challenge, scientifically speaking. How do you reconstruct a detailed picture of an ecosystem from sediment that's 2 million years old? Especially when the obvious stuff like animal bones is mostly gone.
Well, the breakthrough is ancient environmental DNA, eDNA.
Right.
So the team collected 41 samples. These were sediment cores rich in organic matter, taken from different layers in the Cap-Cuban-Have Formation. They weren't hunting for bones. They were after the microscopic stuff, shed skin cells, bits of dung, decaying plants, all mixed into the dirt.
And the age is the kicker here, isn't it?
Oh, absolutely. That's what makes this study such a landmark. They successfully got DNA sequences that are reliably dated to about 2 million years ago.
2 million?
Yeah. And that doesn't just nudge the boundary, it kind of blows past the previous record. The oldest DNA before this was from a mammoth tooth, maybe 1.2, 1.7 million years old. This is significantly older. It's a huge leap in getting genetic data from deep time.
So if you're listening, you might be thinking, okay, how sure are they? How do we know this isn't just like modern DNA contamination? Or maybe it's old, but not that old. How do they actually pin down that 2 million year age for the DNA itself?
Good question. They used a combination of techniques, which is really the key. First, there's the physical geology and something called cosmogenic burial dating.
Okay.
That looks at how certain rare isotopes build up in minerals when they're shielded from cosmic rays, basically telling you how long the sediment's been buried. That gave them a maximum age limit around 2.7 million years, plus or minus a bit. So that sets the outer boundary.
Sort of the no older than this date.
Exactly. But the really cool confirmation came from molecular dating. Think of it like using DNA's own mutation rate as a clock.
A molecular clock?
Precisely. They focused on chloroplast DNA from the birch genus Betula. It's common, and we have a decent idea of how fast its DNA mutates over long periods. By comparing the ancient Betula DNA to modern relatives, they could estimate its age.
And what does that show?
It gave a median age estimate of about 1.3 million years. But crucially, the statistical range, the 95% confidence interval, went all the way up to 2.02 million years.
Oh, okay.
So that independent biological dating lines up really well with the older geological time frame. The different methods basically agree.
So the age seems solid, but then there's the other puzzle, 2 million years. That's an incredibly long time for something as fragile as DNA to survive. Chemically, it should break down.
Yeah.
Right. Hydrolysis, microbes chewing it up. Why did it last so long here in permafrost? What was the preservation secret sauce?
Right. It wasn't just the freezing temperatures, though that obviously helped slow things down. The real key seems to be the sediment itself, the geology.
The dirt.
Yeah, the dirt. DNA does degrade, but it can also stick to mineral surfaces, and that offers protection. When they analyzed the sediment, they found it was really rich in a specific type of clay, smectite.
Smectite clay, okay.
And this stuff is special, has an incredibly high absorption capacity. Think of it as being super sticky at the molecular level, maybe 200 times stickier than common sand grains like quartz.
Like molecular flypaper.
That's a great way to put it. So when the DNA fragments latch onto these smectite particles, they get physically shielded. It makes it much harder for enzymes from microbes to actually get at the DNA and break it down. So it's this combination. The deep freeze of the permafrost plus this super adsorbent clay. That created the perfect long-term storage.
That's amazing. A tiny shield made of clay. Okay, so they cracked open this 2 million year old time capsule. What did the DNA actually show? What was living in Greenland back then? Let's start with the plants.
Well, the plant results were pretty startling. They found evidence for basically an open boreal forest, not the sparse tundra you might expect at 82 degrees north today.
A forest.
Yeah, clear genetic signals for trees like poplar, birch, and even thuja trees like cedars or arborvitae.
Thuja, way up there.
Exactly. And even more telling, they found DNA from plants that today are restricted to much, much warmer, more southerly places. Think North American boreal forests or even northern deciduous forests. Plants like Passia spruce and crataegus, which is hawthorn.
Hawthorn in North Greenland.
I know. Altogether, they found that about 39% of the plant genera they identified, well, they just don't grow anywhere in Greenland anymore.
In this eDNA approach, it revealed much more than the older methods, like looking at pollen grains or larger fossils.
Oh, vastly more. It was far more sensitive. This single study detected 5 times more plant genera than previous eDNA work had found there.
Right.
And get this, 24 of the genera they identified were completely new discoveries for the Cap-Kubenhaven site.
Wow.
Yeah, things never seen in decades of looking at the macrofossils or pollen. So the true biodiversity was just hidden until they looked at the DNA.
Okay, so clearly a productive landscape.
Yeah.
Enough to support a forest, but forests have animals, right?
Yeah.
And you said the fossil record for animals was pretty poor.
Pathetic, yeah. A few bits of rabbit, some insect fragments.
So here's where it gets really wild.
Yeah.
What did the eDNA find about the bigger animals, the megafauna roaming this ancient forest?
This is probably the most jaw-dropping part. The standout finding was the detection of mastodon DNA, specifically mammoth species.
Wait, a mastodon? Like the elephant relative. In North Greenland. 2 million years ago.
Yep. A member of the extinct lineage within the elephant family, found at 82 degrees north.
A mastodon is huge. It's a browser. It needs tons of leafy vegetation every single day.
Yeah.
That doesn't just suggest a forest. It screams productive forest, right?
Precisely. Its presence is probably the strongest biological indicator you could ask for, showing this was a rich, non-tundra environment. They also picked up DNA signatures for ancestors of animals we know today. Things like reindeer or caribou, orangifer.
Okay, that makes sense for the Arctic.
Right. Also hares or rabbits, Leporidae, various rodents like voles and lemmings, Chrysetidae, and even geese, Anatidae.
Interesting.
And one interesting point, every single vertebrate they identified was a herbivore, which actually makes sense from an eDNA perspective.
How so?
Well, large herbivores tend to produce a lot more biological material like dung than predators do, so their DNA is just more likely to end up concentrated in the sediments where it can be preserved and later found. More droppings mean more DNA signal.
Got it. Okay, so we have temperate-associated trees, massive browsers like mastodons. It all points to a dramatically warmer climate. But what was the sort of smoking gun? The clearest evidence that surface temperatures were much higher back then.
For that, we need to look at the marine signals in the DNA. And the really big one was horseshoe crab, Lemuridae.
Horseshoe crab.
Yes. And this is maybe the single most powerful climate proxy they found. Today, horseshoe crabs in the Atlantic. They don't spawn any further north than the Bay of Fundy. That's around 45 degrees north latitude.
Okay, and Cap Copenhagen is at 82 north.
Exactly. So finding horseshoe crab DNA at 82 north strongly implies that the coastal waters back then in the early Pleistocene Arctic were warm enough for them to complete their life cycle, which means radically warmer sea surface temperatures compared to today.
That's incredible. It's like finding crocodiles in Canada.
Kind of, yeah. And they also found DNA from green microalgae species that, again, are only found in more temperate waters today. So the marine evidence really backs up the picture of warmth.
So if you put it all together, you have this ecosystem that just shouldn't exist by modern standards. Temperate North American trees, giant mastodons, warm water crabs, all living together way above the Arctic Circle. It's like a biological mashup.
It really is. A mix of boreal and Arctic species that we just don't see anywhere today.
So what are the big takeaways from this ecological puzzle? What does it all mean for how we think about past climates, and maybe even future ones?
I think the single biggest implication is that this ecosystem, the one they reconstructed at Cap Copenhagen 2 million years ago, it has no modern analog. There's nowhere on Earth quite like it today.
Why not? What made it unique?
It seems to be this unusual combination of three things. One, much warmer temperatures, as we've discussed. Two, the high Arctic light cycle, those really long summer days for photosynthesis. And three, crucially, atmospheric CO2 levels back then were actually lower than they are today.
Lower CO2, but much warmer.
Yeah, likely due to other factors in the climate system back then, like ocean currents or planetary orbit cycles. But that combination warmth, long daylight hours, and relatively lower CO2 allowed these boreal-type species to push much further north and thrive in a way that's just not possible now.
And how is this relevant for us looking ahead?
Well, it gives us invaluable data for modeling future warming. Understanding how that specific environment supported big animals like mastodons tells us something about the potential productivity of high-latitude ecosystems under warmer conditions. It suggests that as things warm up, the Arctic might become capable of supporting denser vegetation and larger animals than we might have assumed.
So it helps us predict how ecosystems might reorganize as climate change pushes species northwards.
Exactly. It gives us a real-world example, a data point from Earth history, showing how complex life can respond to extreme warmth in the Arctic. It's a glimpse into a potential future, informed directly by the deep past.
Okay, that makes sense. Now, we should also talk about, the scientific process. This technique is powerful, but surely there are limitations, right? The things we need to be careful about when interpreting this 2-million-year-old DNA.
Oh, absolutely. Critical thinking is essential here. There are definitely limitations to keep in mind.
Like what?
Well, first, the number of DNA reads you get for a particular species doesn't automatically tell you how abundant that species actually was.
Ah, okay. So lots of birch DNA doesn't necessarily mean the whole landscape was
What?
covered in birch.
Precisely. They found lots of salix, willow, and betula birch DNA. That could just mean those plants happened to grow right next to the stream or pond where the sediment was deposited. Their DNA got concentrated locally. It doesn't prove they dominated the entire region. So presence-absence is strong, but relative abundance is trickier.
Got it. What else?
Second, for some of the really exciting finds, especially the extinct ones like the mastodon, the identification is based on relatively few unique genetic markers, or SNPs, single nucleotide polymorphisms. Basically, small points of difference in the DNA sequence. For the mastodon, the number of informative SNPs they could recover was quite low. So while the evidence points towards mammoth, the confidence level for that specific identification is lower than, say, identifying birch or reindeer, where we have much better modern genetic comparisons.
So the overall picture of a warm forested ecosystem is robust, built on lots of different lines of evidence.
Exactly.
But maybe the mastodon piece, while amazing, is a slightly less certain part of that picture on its own.
That's a fair way to put it. The strength is in the totality of the evidence, the sheer diversity of plants, the other animals, and especially those really strong marine indicators like the horseshoe crab. They all point consistently towards a warm, productive, non-analog world.
It really underscores the power of this eDNA approach, doesn't it? It fills in huge gaps where the traditional fossil record just comes up empty.
It really does. It gives us unprecedented detail about life's distribution in the high Arctic during these really critical warm periods in the past. It's transforming paleoecology.
So the big takeaway seems to be that we need to revise our mental image of the ancient Arctic. Instead of thinking of it as always sparse and icy, we now have evidence for this bustling, open boreal forest community, productive enough for giant animals like mastodons, all the way up at 82 degrees north.
It's a fundamentally different picture.
Which leads us to a final thought, something for you, the listener, to maybe ponder.
No.
If DNA, stuck to clay particles and frozen solid, can survive for 2 million years in Greenland, what other ancient worlds, what other lost ecosystems, perhaps even older ones, are currently locked away in the permafrost around the globe, just waiting for us to find the right technology to read their stories?